My name is Chris Baldessano. I'm an assistant professor in the Department of Psychology at Columbia University, and this is an introduction to using hidden Markov models for event segmentation in neuroimaging. There is a long line of research in cognitive psychology showing that we divide up our continuous naturalistic experience into discrete events separated by event boundaries. These event boundaries have impacts on dynamics during perception. They influence the state of our working memory, as revealed through immediate recall experiments, and they can also change the structure of our long-term memories. How can we identify whether a brain region is sensitive to event boundaries? One possibility is to have human observers annotate the times during a stimulus at which they think event boundaries occur, and they could do this, for example, at a coarse and fine timescale. And then we could look to see whether this brain region shows some kind of response at these human-defined event boundaries. One problem is that different brain regions might respond to different types of event boundaries, and this makes it hard to generate all the different possible hypotheses about where these event boundaries might be. What we'd like to do instead is look directly at fMRI data or other types of neuroimaging data and be able to pull out from this data where the event boundaries are. The basic idea is to identify event boundaries as time points when there is a shift in fMRI activity patterns. For example, if we look at these three voxel time courses, we can identify two moments at which many of these voxels show shifts in their activities, dividing up our time courses into three events, each of which has a characteristic spatial pattern of activity. For example, we can see this type of temporal structure by looking in the angular gyrus during the beginning of the Sherlock dataset. If we take the spatial patterns of activity in the angular gyrus and measure the correlation between all pairs of time points, we can obtain a matrix that looks like this. The blocky structure along the diagonal of this matrix indicates moments when spatial patterns of activity remain relatively stable and then suddenly switch to a new pattern of activity. This is the type of signature we're looking for to define event boundaries. The approach to detecting this structure that I'll describe in this video uses a hidden Markov model, or HMM. The HMM assumes that the brain moves through a sequence of latent states which correspond to the events. It starts in the first state, on every time point either stays in the current state or moves to the next one, and then ends at some final state k. What we get to observe is a sequence of brain activity, these b sub t variables. What we assume is that every event has some characteristic pattern of activity, denoted as m sub k, and that all of the brain patterns during that event should be correlated with that characteristic pattern of activity. So given a sequence of brain activities, b sub t, we can try to infer both where these event boundaries are, so which event every time point belongs to, as well as what these characteristic event patterns are. The way the model does this is by alternating between estimating each of these variables until convergence. This model requires us to choose the number of events that we want the model to find. How can we pick the right number of boundaries? One option is to fit the model on some training group of subjects, and then look in some held out test subject at what the spatial pattern correlations are within and across event boundaries. We can choose the number of events that maximizes this within minus across event boundary correlation. Another approach, which is what we use in this tutorial, is to look at the log likelihood of the model, which is a measure of model fit. We can again train the model on some subjects and measure this model fit on held out subjects, determining the number of events for which this model likelihood is the highest. After identifying event boundaries of this model in different brain regions, we can compare those boundaries to those annotated by human observers. What we've found in general is that regions such as angular gyrus and posterior medial cortex tend to have event boundaries that correspond well with those identified by human observers. We can also look at how the optimal number of event boundaries differs across regions. What we find is a gradient from sensory cortex to high-level cortex, such that regions such as visual cortex and auditory cortex have a large number of event boundaries, whereas in high-level cortex we see much longer events on the scale of up to a minute in length. A recent study by James Anthony and colleagues applied the HMM to data from subjects that were watching basketball games. They found that in V1, event boundaries corresponded well with moments when there was a possession change in the game. They found that in VMPFC, the number of event boundaries that was identified by the model as optimal was related to the amount of surprise in the game. What if we have two datasets with the same sequence of events? For example, watching a movie or listening to an audiobook of the same story. With the HMM, we can find the sequence of event patterns that occurred during movie watching and look to see if that same sequence of patterns appears in the audiobook data. This allows us to find event patterns across datasets with potentially different timings. Using a dataset like this, in which subjects were presented with either a video or audio version of the same story, we found that in some brain regions the HMM could perform this alignment, identifying modality-independent sequences of event patterns. Rather than aligning across modalities, we can use the same kind of analysis to look for sequences of event patterns that are shared between perception and later free recall. For example, this subject watched a 50-minute movie and then spent about 35 minutes freely recalling the events of that movie. The boxes show a human annotation of what part of the movie this subject was talking about during each minute of the recall. By applying the HMM to data from the posterior medial cortex, we're able to find this same alignment without using any of the annotations. This alignment shown in green shows the model estimate of what moments during the movie correspond to each moment during the recall. This type of alignment analysis can also be used to detect temporal shifts between datasets. For example, we find that when a subject watches a movie multiple times, event boundaries can shift earlier in time by up to 12 seconds. We can also find shared sequences of event patterns across different stories if those stories have some similar underlying temporal structure. For example, we can find a sequence of event patterns common to restaurant stories or common to airport stories, and then use these event templates to classify a new story as belonging to either the restaurant or airport schema. We find that we can do this in a number of regions, most strongly in MPFC. This HMM approach has also been applied to other neuroimaging modalities besides fMRI. For example, using EEG, we can detect reactivation of event sequences during free recall. This tutorial will show you how to run these HMM analyses using the Brainiac Python toolbox. There is also an older version of the HMM code that's written in MATLAB, though this version is no longer maintained.